Hello, everyone. First of all, thank you for being interested in my work. My name is Analía Mattiacci, and I'm a postdoctoral fellow at Insects Population Ecology Group in Bariloche, Argentina. We study the behavioral ecology and biology of several insects, some of which are social insects. Social insects like bees and ants are mainly known for the sophisticated colonial organization, which relies on the division of labor, which is the specialization of individuals in reproduction or colony maintenance tasks. Task specialization is typically ensured by individuals of different ages. Underlying mechanisms of age polyism involve nutrition, age, and reproductive status of the individuals. The response threshold model, which has been hugely influential in this framework, suggests that these variables interact to modulate the individual's sensitivity, and therefore the responsiveness to biologically relevant stimuli associated with specific tasks, thus leading to the emergence of division of labor. Vespula germanica is a social scavenger and generalist wasp that in the last century has invaded forested and urban areas around the world. Several studies have suggested that Vespula species exhibit weak temporal polyism, where young workers focus on nursing activities and later foraging specializations of pulp, carbohydrates, and protein develop. However, Vespula species workers do not stop foraging for a given material abruptly when they begin to forage on a new one. It is not clear which are the factors that modulate the decision to exploit resources in wasps in general. We evaluated the interplay between appetitive stimuli responsiveness and task specialization in Vespula germanica wasps. To identify individual responsiveness for appetitive stimuli, we harnessed individually pre-foraging and foraging wasps in a plastic tube that allowed the free movement only of the antenna and mouth parts. Then, we tested the response to stimulation on the antenna for a sequence of increasing concentrations of either sucrose or meat peptone solutions. Then, we quantified the individual sucrose or peptone responsiveness as the total of each wasp's positive response to either stimulus. Assuming a switch in behavior may reflect a change in physiology, we expected pre-foraging and foraging wasps to exhibit a difference in stimulus responsiveness according to the nature of the nutrient. Yet, this was not the case. We can observe from the figures that threshold responsiveness between subcastes is not significantly different. Also, both for sucrose and meat peptone, responsiveness thresholds mostly range in concentrations between 5 and 10%. So, to conclude, modulation of the responsiveness to appetitive stimuli does not seem to be a crucial underlying mechanism of age polyism in Vespula germanica workers. Our results are in agreement with previous works, which suggest that Vespinae could use a generalist strategy of division of labor rather than a specialist one, such as that observed in other social insects. More studies are needed to understand the mechanisms for age polyism in Vespinae. We thank members of the Insects Population Ecology Group for field and lab assistance, statistical advice, and critical discussion. We also acknowledge support from the Instituto de Investigaciones Forestales y Agropecuarias Bariloche, IFAB. This study was financed by the Agencia Nacional de Promoción Científica y Tecnológica and the Instituto Nacional de Tecnología Agropecuaria. Thank you very much for listening to me. You can contact me for questions or suggestions.